Yeah, welcome back, guys. Now let's talk about the etiology of what? Etiology of leukemia. So let's study the causes of leukemia. Now, the study of the causes of leukemia can actually be discussed under three headings. Leukemia can be caused by genetic factor, genetic factor. It can also be caused by environmental factor, environmental factor. Finally, it can also be caused by viral infection, viral infection, viral infection. Now, majorly, the causes of leukemia will always lie within one of these three. Now, the point is, what is genetic factor? Genetic factor mainly is talking about the genetic makeup, so the genes. When some things happen to the gene, the point is, they change, and when they change, they can proliferate. Now, the question is, what does the genetic factor, what does gene has to do with the white blood cells? Remember, we call them blood cells because they are cells. Within a cell, you have a nucleus, and within the nucleus, you have some kind of DNA, and DNA contains things like the gene. So the point is, the gene of these cells, these myeloid stem cells, lymphoid stem cells, inside them they have their own genetic makeup, so chromosomes within them. So the point is, when some things are affected there, that can actually cause what? That can cause things like what? Like uncontrolled proliferation of the cells. Now, for you to understand better, let's just speak a simple cause. Here, under genetic factor, we have things like chromosomal transmutation. Chromosomal what? Transmutation. Transmutation. And the point is, what is the meaning of that? So when they say chromosomal transmutation is from the word chromosome. Let's assume we have a chromosome right here. So just an illustrative diagram. So you have a chromosome right here now. I have a chromosome here. That is a chromosome. You see, inside the chromosome, we have some certain things inside it called genes. Called what? Gene. So inside them, we have some kind of what? Some kind of gene inside them. Now, let's assume we have a different chromosome right here. Another chromosome. Inside this guy too, it has its own what? Its own gene as well. You see, when it comes to transmutation, apologies about that. You can just think of it as transfer. So the point is, in chromosomal transmutation, there's actually a transfer of what? Of gene from one what? From one chromosome. If this is chromosome A and this is chromosome B, there's a transfer of gene from one chromosome to the what? To another. This gene for this chromosome A can move to this one. It can break off and what? And travel to this side and start living with this guy. And when this happens, there has been a change in chromosome A. And that change is what? Lack of that what? Lack of that gene, because the gene has left. And there's a change as well in chromosome B, which is addition of what? Of this gene to this guy here. You see, what we just explained about change means mutation. So when they say chromosomal transmutation, it is just a change that occurs to what? Some chromosome, whereby a certain gene leaves the chromosome and got attached to what? To another. That is what chromosomal what? Transmutation really means. Now, the point is, we have a lot of chromosomal transmutation in human body. So the point is, if we have this chromosome A, if it becomes chromosome 9, that is the 9th chromosome on the what? The 9th chromosome inside a myelogenous stem cell, for example now. The myeloid stem cell rather. We're talking about what? We're talking about the production of white blood cell. So we said it can be myeloid in origin or lymphoid in origin. So either of these cells, inside them, their nucleus, their DNA, they have this chromosome 9. And this B happens to be chromosome 22. You see, inside this chromosome 9, we have a gene there, this certain gene. Its name is ABL1 gene. ABL1 gene. And inside the chromosome 22, this is called BCR gene. Now, when this gene, the ABL gene, when it moves and comes straight up to attach itself, so this guy, when it comes here and attaches itself with what? With the BCR gene, they form a union, and that union is called ABL1 BCR gene, or you call it BCR ABL1 gene. But popularly, we used to call this Philadelphia chromosome. Philadelphia what? Philadelphia chromosome. As simple as that. And the point is, what concerns Philadelphia chromosome with leukemia? When this thing happens, when these two guys, when they fuse together in chromosomal what? Transmutation, the point is that they lead to the production of what? Production of the kind of protein. We call it tyrosine kinase. So there's going to be an increase in the production of what? The protein called tyrosine kinase. And on the long run, let me just jump the gun. When I say jump the gun, as in skip a lot of processes, because it has its own receptors and the likes. We can't be talking about that, or else we are going to get this leukemia wrong. We are going to dedicate a class to oncology generally. And while dealing with the oncology, we talk about all these things, all these mutations. So we have point mutation, amplification, so all this deletion. So everything we talk about them in details. But for now, the point is when there is increase in the production of what? Of tyrosine kinase. What this guy causes is that it causes an increase in the what? Uncontrollable proliferation of what? Of cell. That is the first thing it causes. But one thing about our body is that when a certain cell starts behaving in the wrong way or the wrong manner, the body will look for a way to kill that cell in a process called apoptosis, which is a programmed cell death. Now, when we said if this thing is occurring in the myeloid stem cell, for example, now, let's say we are inside the nucleus of what? Of the myeloid stem cell. The myeloid stem cell. So the point is, this myeloid stem cell is meant to be killed. However, this tyrosine kinase, the first thing it does is what I've already told you, that it causes what? Increase in the proliferation of what? Of that certain cell itself. But the second thing it turns on is that it shuts down, it reduces apoptosis. Apoptosis. When I say apoptosis means what? A programmed cell death. So the point is, when chromosomal transmutation occurs between the chromosome 9 and 22, is an objective question actually. I said this is called Philadelphia chromosome, where the what? Where the ABL1 gene migrates to the what? To the BCR gene inside the 22 chromosome. So they form what? ABL1 BCR or BCR ABL1. So we call them the Philadelphia chromosome. And what they do when this thing connects together is that this leads to the production of the tyrosine kinase, a type of protein. And what it does is that it increases uncontrollable proliferation of cells, and then it's what? It reduces the what? Apoptosis. That cell is not going to revive at that. So as simple as that. Now, this is how what? A certain chromosomal transmutation can cause leukemia. We have other types of chromosomal transmutation. So another one that we have is the PML-RAI. So another chromosomal transmutation is what? Is the PML, so so RAI. So this occurs between the 15th to 17th chromosome. Exactly the same thing that happened here is going to happen here as well. What do I mean by that? The point is just that inside what? Inside the chromosome 15. So you have a gene there called the PML. And then inside the chromosome 17, we have another gene there called the what? The RAI. So this happened. What's happened here rather is that this PML is got attached to what? To the RAI. And when this happens, it leads to another activity I don't want to be stressing you about. What it will cause is actually these two things mainly as well. So what it causes majorly is that, okay, Let me just give you a snippet. When you say RA, they're talking about retinoic acid receptors. So the function of retinoic acid receptors is going to be affected, it's going to be disrupted. And when this happens, what it causes is that it prevents a cell from going into what? From going from a stage to another. What is the meaning of that? If a cell is okay, good. We're talking about leukemia already. You know the stages of what? Of blood divisions. So the point is, you know that myeloblast is going to change to what? It's going to change to something like the promyelocyte cells. So if something is obstructing the growth of that myeloblast into that, it can actually be the effect of the retinoic acid. So as in the retinoic acid receptors. So the point is retinoic acid receptors. So it's actually a stage of white blood cell that I did not give to you called the promyelocyte. But leave it for now, guys. You don't need it to. So in short, what it causes is that it damages the DNA of the cell, and that actually leads to what? It leads to the production of immature cell. So what PML actually causes is production of immature what? Immature cells. Immature cells. Because it stops a cell from moving from immature to what? From moving from immature to mature. Lots of stuff inside it. So because I'm not doing cancer majorly. So the stay tuned by God's grace. I'm going to do oncology on this channel, and we'll explain all this thing in depth. We're going to trash a whole cell. We pick a whole cell like this, and not like our normal year one day is talking about mitochondria power, is talking about things like, okay, let us destroy something. Forget about that. We're dealing with the nucleus alone. We pick it out, we dissect it. What do we have there? Inside that, what do we have next? Inside that, what do we have next? So, talking about oncogenes, talking about the police of the cell cycle. So, all those kind of things. So, but for now, leave it alone. Now, apart from that, we also have other words, other chromosomal transplantation. Another chromosomal transplantation I can think of now is 821. Good. So, 821. So, 8/21. We already know what will happen. The gene of 8 will run towards 21. As simple as that. And that is actually called, I think they run T1 X1, something like that. Let me just put it down. So we have the RUN X1 to RUN X1 T1. Cool. So the point is, for these 8 now, the gene inside it is an RUN X1, run X1. So, and what? Inside 21 is what? Is run X1 T1. So exactly the same thing that happened here and here will happen there. And when that happens, all the lead to is just anything between these three. So production of immature cells, because they prevent them from progressing to the next stage. Uncontrolled proliferation of the cell, and what? They kill apoptosis. So they inhibit apoptosis. So the cells, they're not going to die, they're going to be immature. Now, apart from that, still have genetic factors. We have some other types of mutation that can actually affect the cell. So we're talking about all the cell of leukopoiesis, things like the lymphoblast, lymphoid stem cell, all these myeloblasts. Point mutation can happen to them. Point mutation majorly affect just a single gene most of the time. So not things like changing all over. So a particular cell affected in leukemia is the FLT3. Yeah. So point mutation can actually affect the FLT3 gene. So and a lot of genes like that inside it. So no stress on that, leave it. So another thing is popular thing, trisomy. Trisomy. Another genetic factor, trisomy can actually affect as well. And the question is trisomy. When they say tri means 3. So the point is the some in front of it. We know that some in anatomy is talking about body. When it says somatic cells, body cells. But yeah, code it with chromosome. What? Chromosome. So when they say trisomy, it means a chromosome that's supposed to occur once occur thrice. And an example of that is trisomy 21. Trisomy what? Trisomy 21. The chromosome number 21, instead of occurring in a single place to become the chromosome number 21, simple, it occurs in 3 places. 21, 21, 21. And this trisomy 21 refers to Down's syndrome. Down what? Down syndrome. So Down syndrome. So it refers to what? To Down syndrome. So when you see kids with Down syndrome, what happens to them is actually trisomy 21. So there's an expression of what? Of 2 more chromosomes 21, apologies. So that is what trisomy 21 means. But what does trisomy 21 have to do with leukemia? Because we have 3 instead of 1. It's as if it's giving more surface area, as in an overexpression of what? Of the genes. And when genes are overexpressed, what they cause is just uncontrolled proliferation of cells. As simple as that. Now, that's on the genetic side. We still have a lot of things on the genetic side, but I don't want to disturb you too much. Now, what about the environmental factor? You see, the environmental factor, it revolves around mainly two things. So, the first one is radiation. Cool. So, under this environmental factor, so I'll try to do this. So, under the environmental factor, it revolves around two things. The first one is radiation itself. Radiation. And the other one is chemical exposure. Chemical what? Chemical exposure. Now, what is radiation? And how can it cause leukemia? You see, in radiation, they're talking about some kind of ionizing energy, some kind of ionizing radiation. So example is this is your radiotherapy. Good. Let's take one. So radiotherapy. Radio what? Radiotherapy. You see, radiotherapy is a kind of treatment they give people that have cancer most of the time. So they expose them to some kind of rays. These rays, they have the ability to penetrate the skin, penetrate the bone, and they will go in to make the what? The blastic cells. All these myeloid stem cells, lymphoid stem cells, all these promyelocytes, they can actually go into these cells, damage their DNA. And when they damage their DNA, they will never be able to proceed to the next stage. And that will make them immature. So, mainly, radiation damages the what? Damages DNA. And it can actually cause immature what? Immature cells. Immature cells. But not only that, it can actually cause uncontrolled proliferation as well. The effect of an external factor on the gene will determine whether it is what? Whether it is just damaging the DNA to prevent the what? The production of muscles. Or, the production of mature cells. Or it is actually damaging it to initiate the production of more cells. You understand this concept very well in oncology. Just take some. Now, back to work. The point is, radiotherapy is an example of what? Radiation. So, radiotherapy can actually affect. Another thing that uses radiotherapy, as in radiation mainly, are things like the atomic bomb, the nuclear bomb. So, during this World War II, I think, so a lot of people come down with what? Leukemia. Simply because they're exposed to all these radiations. So, coming from things like bombing and the likes. So, apart from that, talking about chemical exposure. Chemical exposure, you can actually divide that into two as well. So, but don't find more than that. People who are exposed to chemicals like benzene can actually be found in smoke. So, all these cigarettes. So, cigarette smoke. So, if you smoke cigarettes so much, you're actually prone to what? To developing leukemia. So, simply because all these chemicals like benzene can actually trigger the what? The beginning of cancer of the blood. Simple as that. It can damage DNA as well. So, another thing is things like chemotherapy. Chemotherapy is actually a treatment for cancer as well. They expose them to some kind of drugs. An example is alkylating drugs, alkylating agents. So what they also do is they tamper with the DNA of the cell and causes what? Proliferation uncontrollably. So another thing is, even under this benzene of the thing or under this chemical exposure, you can think of things like pesticide. So pesticide. So pesticides. So pesticide as well. So they contain harmful chemicals that can actually trigger what? That can actually trigger leukemia by damaging the cells mainly as well. Now to the last category, which is viral infection. Mainly two viruses have been identified, although there might be more. But mainly two viruses have been identified to be able to cause what? To be able to cause leukemia. So the first one is called the HLTV1 virus. And then the second one is called the EBV virus. So what is the meaning of that? The HLTV is called human HTLV. So it's HTLV. So which means human T cell, L is leukemia, and the V is virus, and the T is type 1, space type 1. So when you hear HTLV1, it means human T cell leukemia virus type 1. So it's actually capable of infecting someone and causing leukemia. And for the EBV, that is very popular, we've known it before, Epstein-Barr virus. Epstein-Barr virus. So, simple as that. Those guys have been implicated when it comes to the development of leukemia. So guys, that's all about the etiology of what? Of leukemia. So just to recap, leukemia can actually be caused by majorly three things: genetic factor, exposure to the environmental factors, and what? And viral infection. So the simplest one, the viral infection, these two guys can actually cause what? Leukemia. I'm talking about the environmental factor. We have things like radiation therapy, chemical exposure, so things like benzene in smoke, and then pesticide, chemotherapy. So the drugs they give to them, alkylating agents. So alkylating agents. So cool. So apart from that, we have the what? We have genetic factor, which is the main contributor. So we said things like mutation can cause it. So point mutation of things like FLT3 gene. We talked about chromosomal transmutation of like 3 different diseases. So, but the most important one should always for an objective question is what? Is the Philadelphia chromosome, which is what? Chromosomal transmutation of the 9th and the what? And the 22nd chromosome. So where the what? The ABL1 and the VCR gene are connected together. Now, we said all these things, they can cause it. They can cause leukemia. But the question is how or why. We explained everything. But if we take note of what these guys are causing one after the other, they are causing what? Increase in proliferation. Which means the cell is going to be what? It's going to be more. And then they are causing immature cells. It means this cell, they are going to be what? Immature. They're going to perform their function. And apart from that, other things, the DNA is damaged, they are immature. So all these kind of things. So you already know, they can also induce what now? Apoptosis. They don't die easily. Simple as that. That's why we always need the therapy again to kill them. Something like that. So down on the side. Now let's talk about the pathophysiology of what? Pathophysiology of leukemia. So, how do I about this?